We move on to the last presentation of this section. It's given by Ted O'Neill from the University of California, Davis. And it's on spent medium analysis to facilitate cultivated meat media optimization. The floor is yours. This is the pointer.
Thank you very much. Thank you very much for the introduction. I'm very grateful to be here today to talk to you all about some of the work that I performed for my PhD. So just a brief background about me and my experiences as they relate to cultivated meat. I recently completed my PhD in food science and technology at the University of California, Davis. I worked in Professor David Block's lab, focused on cell culture media optimization and different approaches as part of the UC Davis Cultivated Meat Consortium. I was fortunate enough to also receive a New Harvest Fellowship along the way, starting in 2019. And I also had the opportunity to found the Davis Alt Protein Project at UC Davis in collaboration with the Good Food Institute. Now that I've moved on from UC Davis, I actually recently started a position at Wildtype in San Francisco as an associate scientist for media optimization. So we're focused on cultivated seafood. And today I'm going to talk about some of the work relating to cell culture media that I performed at UC Davis. I'm going to be wearing that hat today. And just as we all know, cell culture media is still one of the biggest challenges facing commercialization and scale up of cultivated meat. And that's because the animal cells that we're trying to grow are complex and have complex nutrient requirements. So conventional formulations for animal cell culture media often contain upwards of 50 or more components, including animal-derived ingredients, and these are qualities that are not desirable for cultivated meat purposes. So that list is an example of one of the basal media formulations commonly used, DMEM-F12. For more detailed insights into cell culture media, I would point you towards the review article that my group published a couple years ago. In our lab, we like to think about cell culture media optimization in two kind of general approaches, black box and white box approaches. So black box is kind of where we're looking to use advanced modeling techniques, machine learning, artificial intelligence to help us to iteratively test multiple media formulations to arrive at a more optimized media with less cost for the experiments and less effort overall. And white box approaches is where we're actually trying to study the cells that we're growing themselves and learn more about what they actually need to grow in culture so that we can make more informed decisions about how to optimize our media. And we had people working on both of these approaches in the lab. I focused more on the white box approaches. So a big tool that I used for that was spent media analysis, which is kind of self-explanatory and kind of echoes some of the work that was presented earlier today. But for this, we tried to measure the different components in the media and how they're changing over time as the cells are growing in it. So for the analyses that we completed, we measured some key metabolites and nutrients, including glucose lactate-free vitamins, water-soluble vitamins using HPLC techniques in the lab. We looked at the bulk and trace mineral content using ICP-MS. Some of the key growth factor cytokine proteins using immunoassays like ELISA. And then we did some free amino acid analysis using an instrument that utilized capillary electrophoresis mass spectrometry. And actually for that, we collaborated with a company called 908 Devices, which produces this instrument called the Rebel, which is a spent media analyzer that focuses on amino acid analysis, can do some other nutrient analysis as well, some vitamins. But essentially the benefit of this instrument is that it requires very small volume of sample, about 30 microliters or so. It's higher throughput than conventional methods, cheaper, easier to use than the conventional amino acid analysis. So all of the data that I will show for amino acid analysis was obtained from collaboration with 908 devices. So the spent media analysis studies that we performed in the lab kind of fell into these two kind of experimental setups. So the first one was looking at serum versus serum-free media to see if the like a given cell type would differ in its nutrient utilization patterns when grown with serum or with serum-free media. And we basically had C2C12 cells as a model cell type for this purpose and grown in essential 8 serum-free media and PCGM, which is essentially a media that contained 20% serum. And the results of this study were published recently, earlier this year, in Future Foods. The second spent media analysis study looked at 3 different cell types that could be relevant to cultivated meat research and development. So I isolated chicken muscle precursor cells and chicken fibroblasts from chicken embryos, and I compared those with the C2C12 mouse-derived muscle cell line. And these were all grown in a serum-containing media and grown for up to 3 weeks in culture, where I collected samples of the spent media at predetermined time points along that time. And the results, again, are published in NPJ Science of Food. And so I'm going to go through some of the major findings that we obtained, some of the key takeaways of the experiment testing the 3 different cell types. So for the glucose and lactate, one of the biggest points to point out was that the glucose utilization curves differed pretty significantly for the 3 different cell types. So for example, we can see with that pinkish line in the top that the glucose utilization of the CMPCs, or the chicken muscle precursor cells, was not as extensive or rapid as the other 2 cell types, even though they were still growing roughly or similarly to the other cell types, as well as you can see with the purple lines in the lower graphs, which are the cells per well. And keep in mind the right y-axis is a log scale, so that straight line is exponential growth there. So you can see that, for example, the CMFBs, the muscle fibroblast cells from chicken, are continuing to grow even though grow and survive even though the glucose is being depleted pretty significantly towards 7 days in culture. So all of this really is just to highlight that there is in fact a significant metabolic difference among these related cell types that we'll have to keep in mind for optimizing media. So similar findings for the amino acids. Basically, we found that there were actually a lot of amino acids that didn't seem to be utilized very significantly over time by any of the 3 cell types, but there were some that were being utilized, which makes sense. I highlighted them here. And there were also some differences for some amino acids between the 3 cell types. For example, the proline, you can see the black line, the muscle fibroblast cells from chicken, seem to be secreting proline somehow, or it's accumulating in the media, which I'm guessing maybe because the fibroblasts that have produced the ECM collagen earlier on are starting to die off later, past like 7 days, and the collagen is degrading and releasing some proline there. So so again, these are some general considerations that we'll have to keep in mind when we're formulating the amino acid content for our media. Interestingly, when we looked at the water-soluble vitamins and the minerals in the media, they didn't seem to be being used at all by the cells. And more work is going to have to be done to investigate this idea further, but perhaps this means that we can if not eliminate some of these components in the media, at least we can potentially reduce their concentration significantly, or just not really care too much about them, as long as the cells are still happy, which we'll have to test. And then looking at the specific cytokines and growth factors. So first we performed a screen of 30 different cytokines. I'm only showing 5 of them here because these were the 5 that showed more significant differences over time. And so the bars on the x-axis are the time points in culture, basically from the beginning, middle, and end of the culture period. And the y-axis is the concentration of the proteins. And we can see that although the statistical power was very limited for this experiment, so I wouldn't pay too close of attention to the statistical indications there, you can still discern some trends overall. And I would say that probably the most important one of these proteins here is the FGF, which makes sense, the fibroblast growth factor. So we looked at that in a little bit more detail and performed a more extensive analysis of that. And we can see that the FGF concentration decreased very rapidly after the initial seeding. And so that's just going to be something else to keep in mind when we're dealing with these expensive proteins that could be important for things like differentiation down the line. So just to summarize some of the key takeaways of this sort of proof of concept general approach to media optimization, really it makes sense, but you can't really assume that different cell types will have the same media requirements, and so we're going to have to spend some amount of effort and money and time into developing media specific for each cell type that we want to grow. And that said, we can probably benefit from having more robust, easy to use techniques to actually perform this optimization work, whether that's modeling approaches or analytical tools such as that Rebel device from 908 Devices. There are others as well. But otherwise, the major takeaways from some of the data that I showed were that we saw that a lot of the nutrients weren't actually being depleted much, so maybe there's opportunities to target those for reduction or potentially elimination in the media to save costs. And I will also note that given the same cell type and the same media, it's possible that if that system is grown at like a different bioprocess with different parameters, different scales, those different parameters can also potentially affect the metabolic behavior of the cells. So more work will have to be done to investigate that idea further. So yeah, with that, I would just like to acknowledge the organizations that have supported this work: New Harvest, Good Food Institute, National Science Foundation in the US, and 908 Devices. And I would like to thank everyone for your attention. I'd be happy to take any questions.
Thank you. Important work and very insightful. Questions from the audience? There's one over there. Okay, that will be a major. Oh no, that's.
Hello. Yeah. Thank you for your presentation. Just had a small doubt here. Any comments on the detection limits of these components that that you're trying to detect? For example, is there a possibility that was just too low to be detected by the system? Therefore, you do not see a difference before and after cell growth?
Yes, definitely. That's a great point. There were quite a few cytokines and minerals that we probed for, essentially, and we just couldn't detect them. Still, that information is valuable, which it means that the levels are pretty low and probably are not significant. There's always the possibility that even those low levels have some biological effect. Unfortunately, we just don't know. There's probably other ways to assess that, but we just weren't able to do that ourselves. But good point.
Thank you. All right, yeah.
Thank you. It was a lovely talk, and I really like this sort of rational approach to see what's there, see what's being used, and then determine what needs to be added. Just on the actual FGF at the end there, was your system able to tell the difference between consumption or degradation of the peptide? Because there's quite a lot of literature on the half-life for FGF2 being pretty short.
Yeah, that's another great point. No, we did not test that idea specifically. We definitely thought about that. We tested, I believe, just media left in the culture plates with no cells to look at the degradation of the FGF. We definitely saw a decrease. I don't think I was able to show that data today, but yeah, there is some effect of the cell culture actually using some of the FGF, but there's also, I think, more stabilized FGF variations as well. So, yeah.
Great, thanks.
So one question that I had, you mentioned that some of the components are actually not being used, or are kind of stable, and you suggest that they may be eliminated from the medium. However, for vitamins, they typically are cofactors, so they actually should not be used, right, if they are not degraded, but you still cannot miss them. Did you actually look at eliminating some of these factors?
Yeah, great question. We did not test that idea yet. I think others in my lab will start to investigate that idea further. But I would say still that information is valuable, that they're not like degrading over time at least. So that suggests maybe we can reduce the concentrations to some extent to potentially reduce some of the costs associated with them. Whether that's a significant benefit or not remains to be seen. But yeah, that's a great point. I think definitely we should be investigating that idea further.
Yeah, or with feeding strategies, eliminate those from the feeding. Yes. Well, thank you very much.